Thank you for joining me for this edition of Parker Biology. Today we'll be examining membrane structure. The plasma membrane is an 8 nanometer thick boundary separating living cells from their surroundings. Because the plasma membrane is selectively permeable or picky about what substances cross it, it's able to control traffic moving both in and out of the cell. Cells require the uptake of nutrients and elimination of waste, and it's the cell membrane that allows for these exchanges of vital materials. This image is a computer model of a protein channel that allows potassium ions to leave a nerve cell. The potassium ion is represented by the orange dot, the channel is represented by the blank region, and the subunits of the protein are represented by the colorful region surrounding. When we look at the cell membrane, we find that phospholipids are the most abundant lipid in the plasma membrane. We'll also examine the roles of proteins and carbohydrates in membrane function in a little while. When we focus on phospholipids, though, they can be described as amphipathic molecules. Amphipathic simply means that they contain both hydrophobic and hydrophilic regions. Think back to what we already know about phospholipids. Which part of a phospholipid is hydrophobic? That's right, the tails. Which part of a phospholipid then is hydrophilic? That's right, the heads. When we describe the plasma membrane, we're operating under our current understanding, known as the fluid mosaic model. And this tells us that the membrane is fluid or movable, and it has a mosaic or a collection of a variety of different proteins embedded in it. Membranes weren't actually seen by electron microscopes until the 1950s, but chemical analysis back in 1915 showed that membranes are made of proteins and lipids. Scientists studying the membrane reasoned that it must be a phospholipid bilayer. We can see a model of the phospholipid bilayer in this diagram, reminding us again of the amphipathic nature of phospholipids, seeing both the hydrophilic heads along with the hydrophobic tails. Notice hydrophilic because these are polar phosphate groups. They'll interact nicely with the aqueous surroundings of the cell along with the watery contents of the cell, whereas the hydrophobic fatty acid chains will clump together on the inside of the bilayer. Here we see another space-filling model. Again, we've got water molecules interacting with the phosphate heads, fatty acid tails hiding in the middle, and again another hydrophilic head on the inside of the membrane. An early model for the plasma membrane was developed by Davison and Danieli back in 1935, and they proposed what they called a sandwich model. They said that the phospholipid bilayer lies between two layers of globular proteins, so that proteins coated both the inside and outside of the membrane. However, remember the fact that different R groups have different properties? Well, since some amino acids have hydrophilic R groups, but some amino acids have hydrophobic R groups, it didn't really make sense that all the regions of the membrane proteins could be in contact with water. Chemical analysis of membrane proteins showed that they are also amphipathic, so even the proteins have both hydrophilic and hydrophobic regions. So the model clearly needed revision. In 1972, Singer and Nicholson proposed the fluid mosaic model, saying that instead of having the phospholipid sandwich between the proteins, instead the proteins are actually embedded within the membrane. We can see then that this fits much more easily with what we know about chemical composition of proteins. Hydrophobic regions of proteins can comfortably nestle within this hydrophobic region of the phospholipid bilayer, and then hydrophilic regions of the proteins can be in contact with water. The Singer and Nicholson model is supported by freeze fracture studies. Freeze fracture studies allow for the splitting of the phospholipid bilayer, and this has confirmed that proteins are indeed embedded in the membrane. During freeze fracturing, the cell is frozen and literally fractured with a knife. When the membrane is split, each protein is forced to go with one of the layers. The bumps in these micrographs actually show proteins. If proteins were not embedded, we would not see these bumps on the inside surfaces of these membranes. Remember that models are subject to revision as our knowledge increases. This image shows our current understanding of the plasma membrane. Again, the phospholipid bilayer with proteins embedded. We'll look further at the role of proteins and carbohydrates in the membrane shortly. It's important to note that membranes are not static, unchanging structures. Rather, the hydrophobic interactions that hold together the membrane are actually weaker than covalent bonds, and this allows for a lateral or side-to-side movement of lipids and proteins within the membrane. However, molecules do not usually flip-flop across the membrane. Here we can see that lateral movement occurs much more rapidly than the rare flip-flop. The idea of lateral movement within the membrane was supported by Fry and Edinin at Johns Hopkins University. They labeled the proteins on two different cells with two different markers, and then fused the cells. What they were able to see is that over time, the proteins rearranged. So at least some proteins within the membrane are able to move from side to side or laterally. However, some membrane proteins are actually held in place by the cytoskeleton or attachments to the extracellular matrix. In order to work properly, membranes must be fluid. They're usually about as fluid as salad oil, so quite a bit of movement. However, as temperatures cool, membranes switch from a fluid state to a solid state. The temperature, though, that the membrane solidifies at depends on the types of lipids. Membranes rich in unsaturated fatty acids can remain fluid at lower temperatures because their tails have bends or kinks, remember, because of those double bonds? And this prevents tight packing. Another factor influencing fluidity is the steroid cholesterol. This has different effects on membrane fluidity at different temperatures. So at warm temperatures, like human body temperature, 37 degrees Celsius, cholesterol can actually keep phospholipids from becoming too fluid. But cholesterol also hinders tight packing of phospholipids at cooler temperatures. In summary, cholesterol can act as a fluidity buffer, meaning it helps resist changes brought on by drastic temperature fluctuations. Here we can see how the composition of the plasma membrane affects its fluidity. Remember, when we see these unsaturated fatty acid tails, this will prevent packing too tightly and keep the membrane nice and fluid, even at cooler temperatures. However, when we have saturated hydrocarbon tails, the membrane will pack together much more tightly. And this can actually be helpful as temperatures warm up. It can help stabilize and keep the membrane from becoming too fluid. Overall then, cholesterol is going to help stabilize and keep the phospholipids from moving too much, even at higher temperatures, or from packing together too tightly at lower temperatures. Some species appear to be able to change the composition of their phospholipid bilayer depending on environmental conditions. For example, some fish that live in extremely cold environments have a high percentage of unsaturated fatty acid tails in their plasma membrane, and this prevents their phospholipids from packing together too tightly, which allows their membranes to stay fluid. Also, when we think about the ability to change from season to season, wheat plants are subject to drastic temperature changes throughout the year, but in autumn, the percentage of unsaturated phospholipids actually increases, and this helps prevent their membranes from packing too tightly in cold winter climates. On the other hand, archaebacteria, or the thermophiles that live in hot springs, have unusual types of lipids that actually prevent excessive fluidity. Now that we've addressed the fluid part of the fluid mosaic model, let's examine what we mean by the mosaic aspect of cell membranes. Different cells have different proteins, which allow for a variety of different functions. Proteins can be either peripheral or integral. Peripheral proteins are simply bound to the surface of the membrane, whereas integral proteins can actually cross the membrane. They're embedded somehow in the membrane. Here we can see an integral protein, specifically transmembrane, because it does cross the whole membrane. Remember back to what we know about proteins. We can see the N-terminus where the amino group is present. We can see the C-terminus where the carboxyl group is present. Notice that this protein is amphipathic, possessing both hydrophobic and hydrophilic regions. Now, when we think about proteins, we know that they can do a number of different jobs, and the same is true of membrane proteins. Notice that membrane proteins can function in transport of materials, they can serve as enzymes, they can help receive signaling molecules and function in signal transduction, which we'll learn about later in the year. Membrane proteins can help alert cells to other types of cells around them and function in cell-to-cell recognition. They can also help cells join together and even attach to the ECM. Notice that one of the functions we just mentioned was cell-to-cell recognition. Cells must be able to tell different types of cells apart, to recognize neighboring cells and to recognize self versus non-self, especially when we think about our immune system being able to recognize invaders. Now, a lot of times, cell-to-cell recognition is carried out by carbohydrates that are present in the membrane. Now, if a membrane carbohydrate is bonded to a lipid, we call it a glycolipid. However, if a membrane carb is joined to a protein, we call it a glycoprotein. The great diversity of carbohydrates allows them to serve effectively as markers, distinguishing one cell type from another. The idea of surface substances alerting surrounding cells as to their identity is really evident in this HIV study. Now, despite numerous exposures to HIV, a very small number of people do not actually develop AIDS. Scientists have found that these resistant individuals actually lack the membrane component CCR5. These are co-receptors. So in normal people, these co-receptors are required for HIV to bind and infect. However, in individuals who seem to be resistant to the virus, they're lacking CCR5, and the HIV is unable to bind and unable to infect. So we can see then that these surface markers are extremely important. Membranes have distinct inside and outside faces. This sidedness of membranes has to do with how the plasma membrane is built by the ER and the Golgi apparatus. In this picture, we can see that membrane proteins and lipids are synthesized in the ER. The carbohydrates, the little green molecules, are added to the transmembrane proteins, turning them into glycoproteins. Remember, carbs attached to proteins. The carbohydrate portions may then be modified. Then, as we see these substances move over into the Golgi, the glycoproteins undergo further carbohydrate modification, and the lipids grab onto more carbs. The glycoproteins, the glycolipids, and other secretory proteins are then going to be transported via vesicles over to the plasma membrane. When the vesicles fuse with the plasma membrane, the outer face of the vesicle becomes continuous with the inside face of the plasma membrane. This releases the proteins from the cell, a process called exocytosis. It also positions the carbohydrates of the membrane glycoproteins and glycolipids on the outside face of the plasma membrane. So here we can see, because of this fusion, we're now going to have our glycolipids and our glycoproteins attached to the right side of the membrane. That's going to do it for our discussion of the membrane structure. Stay tuned for our next edition when we talk about membrane function. Thanks again for joining me at Parker Biology.